Chapter 8, Sex Selection, Genome Selection, and Designer Babies. In this chapter, we'll talk about all of that. Okay, first of all, saging. Filipino folks believe that kambal na saging, if at women ate a kambal saging, to have kambal na anak. Okay? Yun yung paniniwala ng ibang tao dito sa Pilipinas. Next is the Chinese lunar calendar. Chinese lunar calendar is parang malalaman natin doon kung boy o girl yung magiging anak niya sa araw na yun or sa day na yun. Blue for male and pink for women or for girl, diba? Medyo sexist yung calendar na yun pero ganun talaga. And next is the medyo is sex position. Sex position. Okay, malalaman natin doon. Sabi, Filipino folks or people from the West din siguro na different sex position will lead na siguro yung babae pag lalaki nakapatong sa lalaki yung magiging anak, pag yung babae nakapatong sa lalaki, magiging babae yung anak. Parang something like gano'n, di ba? Yung mga ganong folklore tayong na parang pinapaniwalaan pa. Kung hindi nyo pa naririnig, kung naririnig nyo yan, gano'n yan. Okay, now let's take a look kung ano pang sinasabi dito sa Chapter 8. In vitro fertilization or IVF. IVF is a method of assisted reproduction in which an egg and sperm retrieved from the biological mother and father respectively are fertilized in a laboratory dish. In vitro literally means in glass. Fertilization is a union of sperm and an egg cell resulting in the creation of an embryo. Depending on successful cell fission, the embryo is transferred and implanted into the woman's uterus a few days later. Either the biological mother carries her child or surrogate mother may do so. Usually multiple embryos develop and sometimes more than one embryo is implanted to increase the chances of successful pregnancy, which may result in twins, triplets, or more.
Genetic screening or PGS is the screening test that analyzes the chromosomes of cells from embryos created during IVF before they are transferred to the uterus. PGS only screens for the number of chromosomes and is used to identify chromosomal abnormalities such as trisomy 21. It is also possible to identify the sex, as the sex of a child is determined by the shape of the sex chromosomes, XX for girl or XY for boy, using PGS. In this module, we discuss the ethics of PGS in terms of sex selection.
Implantation Genetic Diagnosis or PGD. PGD is a test in which a cell is removed from an IVF embryo and tested for specific genetic mutations associated with disease or disability before it is transferred to the uterus. PGD is usually performed when either of the parents is a carrier of a certain genetic condition and thus desire to implant an embryo that will not be affected. Very occasionally, parents use PGD in order to select embryos with a mutation associated with what may be societally deemed a disability. In this module, we discuss PGD in terms of selection against or for genetic mutations associated with disease or disability. It is important to note that PGD is only able to investigate and identify existing genes. It does not alter DNA.
Eugenics, defined as a science of improving a human population via manipulated breeding to increase the occurrence of desirable heritable characteristics. Breeding is controlled as opposed to uncontrolled when those with desired traits are encouraged to reproduce with others who have the same desired traits. Those with undesirable traits are prevented from reproducing or even sterilized. Eugenics was the root ideology that caused many historical atrocities, including the Nazi persecution of Jews and the disabled, in which many millions were killed and sterilized.
Breeding may also be controlled by selection of some embryos rather than others or by using genome editing.
Genome editing. Genome editing is the use of technology to alter the genome. Alteration can be done via adding, replacing, removing, and splicing DNA bases.
Designer baby, a child whose genome was engineered or edited in order to express a particular phenotype or eliminate a certain mutation associated with disease or disability. This is in connection with the pre-implantation genetic diagnosis or the PGD. Again, PGD is to screen embryos that are generated through in vitro fertilization. With the use of PGD, doctors can identify embryos that may carry the defective gene, and this defective gene can be altered na nagre-result na nga sa genetically modified baby. Such infants are called designer babies since they have been designed through gene alteration. The primary aim of creating designer babies is to avoid their having heritable diseases coded by mutations in DNA. For example, kapag ang isang pamilya ay may sakit na pwedeng mamana, pwedeng may panganak yung baby nila without that particular disease. Or in short, they want to edit the DNA of their embryos to prevent their future children from suffering. The characteristics people tend to associate with designer babies are intelligence, height, and athletic ability. Last one is the germline engineering. It is the use of genome editing techniques to alter sperm, eggs, or embryos such that the genetic changes become permanently encoded in the sex cells of the resulting adult and are able to be inherited by future offspring. Ginagamit din ito to produce designer babies. It just targets the genes in eggs, sperm, or very early embryos. Ginagamit naman ito kung gusto mo pumili ng gender na magiging baby mo. A way of using genetic engineering for sex selection is then to put the gender genes of the desired sex in a virus-like organism and use germline engineering to alter the sex of an embryo. May similarity din sila ni heritable genome editing because it also involves making genetic modifications to gamete, precursor cells, eggs, sperm, or early-stage embryos in the laboratory.
Sex selection pros: avoid sex selection, abortion, infanticide, and child abandonment. Family balancing, prevention of sex-linked disorders, and parental autonomy.
Selection cons. Promotion of sex selection legitimizes policies that have led to sex selection abortions, infanticide, and child abandonment. Justice concerns a slippery slope, and it changes the relationship between parents and children.
Disability. Pros. Selecting against disability avoids hardship for families. Parents ought to give their children at the best chance at having a life worth living. Helpful to families living with disability to have a child with the same one. Example are deafness and dwarfism.
Disability. Cons. Expressivist argument. Second, problems surrounding disability are caused by society, not by the disability itself. Third, reduced resources for the disabled community.
Moving on to the pros of genome editing. First one is parental autonomy. We already greatly influence and mold our children. Genome editing is no different. Ability to eradicate some genetic diseases and inherited conditions. Equality of opportunity. Human rights approach. Lastly, parents ought to provide for their children a life worth living.
Here are some of the negative claims with regards to the use of genome editing. The child is unable to consent to the life-altering procedure. Propagation of inequality. Child's right to an open future. Lacks of individuality. Last one is the sanctity of nature.
And again, thank you for your time and for listening. This is Chapter 8, Sex Selection, Genome Selection, and Designer Babies.